There's no attendance this morning. We filled it up on Wednesday, and I forgot to bring the new sheet down from my office. So we're in Roman numeral 9, anatomy of a muscle, from the gross structure down through the bands. So the gross structure is literally just, you remove the skin from a part of the body and look at the muscle tissue. And what we do is we go deeper and deeper and deeper until we're looking at things with an electron microscope. And we actually, this is a photomicrograph, obviously a diagram. We actually see the bands and begin to understand how they actually work at the molecular level. So once again, we begin here with peeling the skin off of the arm. In this case, these pictures are from your book. There are one or two I'll show you that are from another book in case you want to snap a picture of them or something. But when we remove the skin from a part of the body, we take one of the muscle. This would be one of the extensors. We follow the arrow. We bring it down here. We blow it up a little bit. We see that there's an outer boundary covering the muscle, and it's called the epimysium. So this white outer boundary, which is the connective tissue, is called the epimysium. Now your book actually fails right here because it doesn't show the true outer boundary of the muscle. So if you have your book, you can add it. If not, you can add it when you get home. But superficial to that epimysium is the real outer boundary. And you know this because you've studied histology, the fascia. So the outermost boundary of the muscle is the fascia. One layer deep to the fascia is the epimysium. The fascia and the epimysium are extremely important because they are how the muscle is connected to a bone. They are also holding together the subunits of a muscle. And as we go through this, what we'll have about three times, is we'll have some outer boundary holding together subunits. It's as though these markers are the subunits, and my hand is the outer boundary holding them together. And then we'll go deeper, and we'll see more subunits. And we'll see an outer boundary holding them together. And then we'll go deeper, and we'll see more subunits and an outer boundary holding them together. And then we'll go deeper. And so once again, if we go back to where I started, the title here, Roman numeral 9, anatomy of a muscle, from the gross structure down through the band. Gross meaning you can basically see it with the naked eye. The gross structures, we've just skinned the animal. Whether it's your arm, your leg, or a deer in the field. We've just skinned the animal, and we're looking at muscle tissue. And we begin to magnify, magnify, magnify. We go through many, many pictures. And eventually, we're looking at the actual bands that can begin to understand how the muscle works at the molecular level. So the outermost boundary, as I've said, is the fascia. Your book fails here because it does not put the fascia in this picture. We've added it, or you will when you get home. Again, the epimysium and the fascia are very important for two reasons. Number one, they are how the muscle is attached to the bone. The second reason that the epimysium and the fascia are so important is that they are holding together these subunits. Right here, they've taken one subunit and pulled it out. those subunits are called fascicles. We're going to go to a picture that's not in your book, where the fascia is shown, and we can start to see how the fascia and the epimysium connect the muscle to the bone. So here's a muscle. These are the subunits of the muscle being held together by the fascia and by the epimysium. And they use a slightly different word. It means the exact same thing, fasciculus. A fasciculus and a fascicle are the very same thing. But if you focus right here, you'll see that the fascia of the muscle is the same thing as the periosteum of the bone. So you learned in histology that ligaments connect one bone to another or hold bones together at joints. You learned in histology that tendons connect muscle to bone, but nobody ever asked me how. Are they duct taped? Are they stapled? Well, the answer is right there. The fascia of the muscle is the same thing as the periosteum of the bone. So let's draw a picture that's even better than either one of these that really shows that. Muscle, bone. So the outermost boundary of the muscle is the fascia, and the fascia surrounding the muscle is continuous with the periosteum surrounding the bone. That's what the tendon is. Not completely, but partly. So literally, the fascia extends through the tendon, is the tendon, and is continuous with the periosteum of the bone. Just deep to the fascia, as we just saw in the other pictures, is the epimysium. The epimysium extends into and contributes to the structure of the tendon, but does not continue and does not surround the bone. As we go through this, if you find it redundant, that's good. That's what I want, because before we're finished, we're going to draw a picture of our own and you're going to label it. So once again, the outermost boundary of the muscle is the fascia. Just deep to the fascia is the epimysium. The fascia extends through the tendon, is part of the tendon, and is continuous with the periosteum of the bone. The epimysium extends out into the tendon, is structurally part of the tendon, but stops there. As you can see in this picture, it does not surround the bone. The fascia and the epimysium, working together, are binding and holding together the subunits of the muscle. We'll have subunits after subunits after subunits, as I've already indicated. There's a picture on Facebook that you've probably encountered by now. If not, you'll want to. But it's a diagram, kind of like what we're going to draw, except it was done with a computer, and so the circles are nice and round, making it much prettier. Make sure that you have that. So we've added the fascia to this picture. The fascia and the epimysium are holding together these subunits, all of those, which are fascicles. So once again, they pull one fascicle out, follow the arrow, bring it over here. So this is one muscle fascicle. This big thing here could be this one or this one or this one or this one that they pulled out a little bit. Now when we focus on the fascicle, we see that the fascicle has an outer boundary, the perimysium. So the perimysium is to the fascicle what the epimysium is to the muscle. Just as the epimysium is holding together the subunits of the muscle, which are fascicles, the perimysium is holding together the subunits of a fascicle, which are muscle fibers. So each one of these is a muscle fiber. I'm sure you remember from last week that a muscle fiber is the same thing as a muscle, your turn, cell. Muscle fiber, muscle cell are interchangeable. They are the same thing. We see our peripherally located nuclei, one of the characteristics that we gave to skeletal muscle last week. So the outer boundary of the muscle, fascia, then epimysium, both forming the tendon, the fascia going all the way through the tendon, being continuous with the periosteum of the bone, the fascia and the epimysium working together to hold together the subunits of the muscle, which are fascicles. And we blow up one fascicle, we see it has an outer boundary called the perimysium. The perimysium is to the fascicle what the epimysium is to the muscle. The perimysium is holding together the subunits of the fascicle, which are muscle cells. If we zoom in a little bit on a fascicle, we see that a fascicle has an outer boundary called an endomysium. So whether we're thinking about a muscle or a fascicle, either way, there's an outer boundary. My hand is the outer boundary. The subunits are fascicles. Now my hand is the perimysium. The subunits are cells. Now my hand is the endomysium. The subunits are, well, I haven't told you yet, but they're myofibrous. So let's blow up. And again, we sort of follow the arrows, although I don't see an arrow here, but it's going to be down that way. So let's blow up a single muscle cell. So here's our single muscle cell. In other words, this is one of those just blown up. The outer boundary of the muscle cell is the endomysium. Now that seems to contradict Biology 111. Because I know when I took it, I learned that the outer boundary of any cell is a cell membrane. Well, remember, aren't there two outer boundaries surrounding the entire muscle? The epimysium, and what's superficial to the epimysium? Fascia. There are two outer boundaries surrounding the cells. There's only one outer boundary surrounding a fascicle, the perimysium. Two outer boundaries surrounding the muscle, the fascia, not in this picture, we've been through that, deep to that, the epimysium. One outer boundary surrounding the fascicle, the perimysium, but when we get to the cellular level, there are again two outer boundaries. The superficial most outer boundary of the cell is the endomysium. Deep to the endomysium is the cell membrane, but they changed the name of the cell membrane. Now it is called the sarcolemma. The sarcolemma is the plain old Biology 111 cell membrane. It just has a new name. Only for skeletal muscle. So if the sarcolemma is the new name for the cell membrane, what do you suppose the sarcoplasm is? The new name for the cytoplasm. And I don't think it's in this picture. It's not. But what would the new name be for the endoplasmic reticulum? Sarcoplasmic reticulum. So instead of an ER, there is an SR. Okay? They didn't rename anything else. Just those three. Cell membrane is sarcoplasm, cytoplasm, or cell membrane is sarcolemma, cytoplasm is sarcoplasm, ER is SR. We can see that there are subunits within the cell. We had the whole muscle, the subunits are fascicles. We had the fascicle, the subunits are cells. Now we look at a cell, the subunits are myofibrils. There's the word right there. The subunits of a myofibril are, I hadn't told you that one yet. Is a myofibril going to have subunits? Sure it is. Yeah. So the subunits of the muscle are fascicles, the subunits of a fascicle are cells, the subunits of a cell are myofibrils, the subunits of a myofibril are myofilaments. As you think about this, you can imagine why we were not able to understand how things really work before we had microscopes. And we could only see the gross structure. And I'll remind you of what the Egyptians did. They thought the brain was garbage. They pulled it out through the nose of the dead before they buried them. Thought it was trash. They didn't have microscopes. They had no way to really understand things. Everything, whether it be physiology or disease or a drug, there's always a molecular basis of how it works. You've heard of the myofilaments, myosin and actin, these so-called contractile proteins of muscle tissue. Myosin is known as the thick filament, and actin is known as the thin filament. So each of these myofibrils, if we had more magnification, and we will in a minute, each of those myofibrils can be subdivided into myofilaments. Literally, if we enlarge that, I know it blurs out, but each of these myofibrils is composed of myofilaments, two different ones, actin and myosin. We just need more magnification to be able to see them, and we'll achieve that momentarily. So the outermost boundary of a muscle is the fascia. Just deep to the fascia is the epimysium. The fascia and the epimysium work together to hold together the subunits of the muscle, which are fascicles. The fascia and the epimysium also form the tendon, which connects the muscle to the bone. The fascia extends all the way through the tendon and is continuous with the periosteum of the bone. The epimysium contributes to the structure of the tendon, but stops where the tendon joins the bone. It does not surround the bone. Each fascicle has a subunit called the perimysium, which binds and holds together the subunits of the fascicle, which are muscle cells, also called muscle fibers. Each muscle cell or muscle fiber has an outer boundary called the endomysium. Just deep to the endomysium is the cell membrane, which has been renamed and is now called the sarcolemma. The endomysium and the sarcolemma are holding together the subunits of the cell, which are myofibrils. Each myofibril has subunits which are myofilaments, two of them, actin and myosin. Myosin is thick, actin is thin. New picture, more magnification. So this is a single cell, really no more magnification here, but want to remind you of the vertical striations, which are not really there. We see the peripherally located nuclei that we learned about in the histology of skeletal muscle. These are the individual myofibrils. So now we blow things up more, and we'll see the subunits of a myofibril. We know this is a cell because they've labeled the cell membrane for us. Each of these is a myofibril. They've taken one myofibril and pulled it out, and now in cross section right here we can see the individual myofilaments within the myofibril. We can also see the myofilaments longitudinally through here. Which myofilament is this one that they're calling thick? And this one is active. Okay. So now we take a little break and we draw our own picture. This is a picture that is on Facebook as a much prettier diagram than anything I can draw. Nasty game last night, huh?
Yeah.
Like you have to incomplete that interception, that would have been a different point.
I'm done with the Broncos. I'm changing teams.
I mean, I don't mean to brag, but don't be a fair weather fan. Come on.
Simeon just needs a few years to put it together. He's young. Oh my God. Feeling that stuff. I'm like fuzzy now. Did Chantel Sullivan ever come in? Is this the Facebook picture? So this is the Facebook picture I'm talking about. It's this, it's just much prettier because it was done with a computer and everything's nice and round. All right, so this is a cross section of a muscle. I'll go first. Fascia, outermost boundary. Fascia is the one, remember, that we had to go to picture from the other book. Show it again because I know a couple of people trickled in, which is fine. I just want you to see it if you didn't. So your textbook shows the epimysium as the outermost boundary. It's the white ring here. This is the fascia, which is the true outermost boundary. You know that from histology. The fascia, as you can see, is continuous through the tendon with the periosteum of the bone. The fascia of the muscle and the periosteum of the bone are the same thing. All right, so what is the green ring? If the black ring is the fascia, what's the green ring? Epimysium. And the fascia and the epimysium are binding and holding together the subunits of a muscle. So these subunits of the muscle are what? Fascicles. Each fascicle has an outer boundary. What's that outer boundary called? Perimysium. So the perimysium holds together the subunits of the fascicle, just as the fascia and epimysium hold together the subunits of the muscle. Perimysium holds together the subunits of the fascicle. What do you call these subunits of a fascicle? Muscle fibers, or what else?
Cells.
Okay? And each muscle fiber or muscle cell has two outer boundaries. The outermost boundary is the endomysium. And deep to the endomysium is the cell membrane, but we don't call it that. What do we call it? Sarcolemma. And we'll see here in not very much longer what all this sarco stuff is about. Sarcolemma, sarcoplasm, sarcoplasmic reticulum. So just as the fascia and the epimysium hold together the subunits of the muscle, which are fascicles, and just as the perimysium holds together the subunits of a fascicle, which are cells, the endomysium and the sarcolemma hold together the subunits of a cell, which are, what are these things? Myofibrils, okay. And the subunits of a myofibril are what filaments? Myofilaments. How many myofilaments are there? What are their names? Actin and myosin. How do you know which is which? How are they different?
Myosin is thick, actin is thin.
Perfect. Myosin is thick, actin is thin. So one last time, I urge you to get the Facebook picture, print it, label it. So we started here, we peeled the skin off of an appendage, just like skinning an animal in the field. We see muscle. Outermost component there is the fascia. Just deep to the fascia is the epimysium. Fascia and the epimysium are structurally the tendon. Fascia specifically runs all the way through the tendon and is continuous with the periosteum of the bone. The fascia and the epimysium together hold together the subunits of the muscle, which are fascicles. Each fascicle has a single outer boundary called the perimysium. The perimecium holds together the subunits of the fascicle, which are muscle cells or muscle fibers. Each muscle cell or muscle fiber has an outer boundary called the endomecium. Deep to that, it has a cell membrane called the sarcolemma, holding together the subunits of the cell, which are myofibrils. Each myofibril is subdivided into myofilaments. When we increase the magnification sufficiently, zooming in on a single cell, we know it's a cell because we see the cell membrane there. We see the individual myofibrils, and now enlarged enough to see the myofilaments within the myofibrils. These bands that we saw, these are not bands, these striations that we saw that aren't really there, now we can understand why we see them. When you prepare tissue to study under the microscope, you have to stain it. Now, the words stain and dye are interchanged all the time, and I guess it doesn't matter as long as one knows really what the difference is. But you use a dye to stain something, even though in your little bottles in microbiology they call them use this stain, use that stain. No, technically they're dyes, and the dyes are used to stain the tissue. It's kind of like using a dye to stain fabric. You don't use a stain to stain fabric, you use a dye to stain the fabric. Anyway, that's a technical difference, and be aware of it, but everybody does interchange the words. Nevertheless, when you stain a tissue to look at under the microscope, depending on what you want to see, you use certain types of dyes. Well, actin and myosin are proteins, and so you're using a dye that binds protein. Well, if you have a piece of tissue and you pour a dye on it, a dye that's going to bind protein, the more protein there is, the more protein there is to bind the dye, the more protein there is to stain, and the darker that area is. So an area like this stains very dark because it contains both myofilaments. An area like this stains darker than an area like this, even though this contains only one filament and this contains only one filament. Why would this stain darker than this? Because that's the thick filament, that's the thin filament. So when we see these patterns like this, it's because of the way the filaments overlap. There's really, there are a couple of things, but these vertical stripes that we see are actually where the actin and myosin overlap. Those vertical stripes are this area right here, where the actin and myosin overlap. So the vertical striations are just an illusion. Now, nobody wants to say the part where we have just actin, or the thin filament, the part where we have just myosin, or the thick filament, the part where we have actin and myosin overlap, that's just too much. So people impose labels. It's just like nobody wants to say the time during nuclear division, when the nuclear membrane breaks down, the DNA coils, and the coil configuration is held together by proteins called histones, and the chromatin becomes visible as discrete chromosomes. We just say prophase! Nobody wants to say the time during nuclear division where the double-stranded chromosomes split at the centromere, and each single strand of the double-stranded chromosome, now a single-stranded chromosome, moves toward, and we just say anaphase. So we impose these labels so that we can have a conversation about them. Well, we've done that with all of this. Let's back up. So wrap your brain around that. We're going to just blow that up, make it a little bit bigger, but we're going to put labels on it. Here we go. So we've got, just ignore the green. The green is a protein called titin. It's kind of like a shock absorber. Just try not to even see it. In fact, we'll do it another way. Got a picture that doesn't have it. All right, so we've got this area where, which filament is this? How do you know it's myosin? Because it's thicker. This is actin. So we've got this area where they overlap. We have this area that's myosin only. We have this area that's actin only. Okay, so this is what I want you to think about. The green one, titin, is not there, but I want to use the one that's in your book because that's what you're going to have when you get home. The units, as we go from left to right, are called sarcomeres. And that's where all that sarco terminology comes from. Sarcoplasm, sarcolemma, sarcoplasmic reticulum. The sarcomeres are to a muscle what the alveoli are to a lung, or what a nephron is to a kidney. The sarcomeres are the functional units, the contractile units. The sarcomeres are the microscopic functional contractile units of a muscle. The boundaries of a sarcomere are Z lines. That's a Z line, and that's a Z line. The A band contains actin and myosin. So look at the boundaries of A from here to here. So within the A band, we have actin and we have myosin. But within the A band, we have the H zone. So the H zone is within the A band and is myosin only. It's kind of funny, they gave everything a nice simple letter, except so we've got the H zone within the A band. The other part of the A band where actin and myosin overlap, look what they called it. The zone of overlap. Why didn't they give that one a simple letter? I don't know. So the area or zone of overlap, so once again, that is within the A band. And is where actin and myosin overlap.
Why do they call it A and H?
No idea. There's another area that you can see called the M line. The M line is within the H zone and is a protein called myomycin. We'll see it enlarged in a bit. So we've accounted for everything within the A band, but remember the sarcomere goes from one Z line to another. The sarcomere is the functional contractile part. Well, this region from the Z line to the end of myosin, and you have the same thing on the opposite side, is called the I band. I band. Z line to the end of myosin. We're going to see the Z line, or rather we're going to see the I band labeled two different ways. Notice that in this picture, the I band goes from the, and this is a mouthful, I know. The I band goes from the left side of myosin of this sarcomere all the way through the Z line that separates this sarcomere from this sarcomere to the right side of myosin in the adjacent sarcomere. Another way to label the I band is to say that's the I band of the sarcomere on the right, and that's the I band of the sarcomere on the left. It's the same thing. It doesn't matter whether we say there's one I band and it spans two sarcomeres with the Z line in the middle, or whether we say the sarcomere on the right has an I band and the sarcomere on the left. It's the same thing, but you're going to see it labeled both ways as you take different classes like patho, medical, surgical, nursing, and all this stuff comes back up. Ugh. Again, the sarcomeres are the functional units, the contractile units. The reason a muscle contracts is because all of the sarcomeres shorten. The reason a lung expands when you inhale is because all of the alveoli expand. If you take my advanced class, 203, where you do the dissection of the cadavers, I'll stick a tube down the trachea of the cadaver and blow in it, and you'll see the lungs expand. They blow up just like balloons because each individual alveolus blows up like a balloon. Now let's compare, let's think again about using the dye to stain the tissue. Let's compare the diagram to the photomicrograph. So these super dark lines here are the Z lines. But remember, those are not the stripes that we see. When we look at this type of picture, this is low magnification compared to this. So these stripes that we see here, those are not the Z lines. That's where actin and myosin are overlapping. So Z, 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 Z. From here all the way to here is the A band. This part of the A band is the area of overlap on the left. This part of the A band is the area of overlap on the right. And as I'm pointing it out here, you can see the labels up there. this, and it's very fuzzy up this close, this part of the A band is the H zone where we have just myosin. And then this vertical line right there is the M line. And then the parts that are super light are the I bands. And so again, we can say this I band belongs to that sarcomere, and this I band belongs to that sarcomere. Or we can say, no. The I band spans two sarcomeres with the Z line in the middle. It doesn't matter. You'll see it both ways. Now, people wondered how the sarcomeres shorten after they figured out that they did shorten. Sarcomere on the left is relaxed. Look at the width of the H band. Sarcomere on the right is partially contracted. Look at how much the H zone is much narrower. Also notice that the I band is much wider here and here. That is the sarcomere on the left versus the I band on the right, or the sarcomere on the right. The I band is much narrower. So they realized that the sarcomeres are shortening because the Z lines slide in, and if the Z lines slide in, the I bands get narrower, and if the Z lines slide in, then the H zone within the A band gets narrower. Let's have some other pictures of that. So we've got one of those I don't want you to see yet. We've got relaxed and we've got contracting. Look at the difference in the I bands. Look at the difference in the H zone. So literally, I know I'm not praying, the Z lines are sliding in. And these Z lines can slide in until they butt up against the end of myosin. They can't go past the end of myosin. Finally, let's look at this as a photomicrograph. So we've got one sarcomere from here to here, relaxed, contracting, more contracted down there. The I band, look at the width. Now look at the width of the I band. Look at the width of the H zone within the A band. Look at how much narrower the H zone is. But watch what truly happens. So as these Z lines are moving in, and this H zone is getting narrower and narrower and narrower, the actin myofilaments don't run into each other and stop. They overlap. If you go back to using a dye to stain a tissue, we've got this super dark area where they overlap, super light area where it's actin only, intermediate area where it's myosin only. What's going to happen here? It's going to be really dark. Yeah. We've got relaxed at the top, contracting in the middle, fully contracted at the bottom. There's your I band. I band is narrower. Look, I don't even see an I band here. That's because the Z line has moved all the way up against the myosin myofilaments. It can't go any further. H zone, that width, H zone is narrower. Now look. See how that little area right there is darker? That's where the actin myofilaments are overlapping. There's your M line. So as we understood that the filaments, or rather the sarcomere shortens, we had to wonder why, how, what's going on here? just like with DNA, if you go back to the cell cycle. When you look at a typical cell under the microscope, you see a nucleus. The DNA is uncoiled. It's long, skinny, thread-like, invisible. We call it chromatin. And during prophase, the DNA goes from this long, skinny, thread-like, invisible, literally DNA molecule, to tightly coiled, held together by proteins called histones. And the DNA becomes visible as discrete chromosomes. How does it go from long and skinny to short and thick? It coils. So, what happens with muscle tissue? Don't know. We had to figure it out. And what we figured out is that, and we can see it in the picture here easily, the filaments slide. They slide back and forth upon each other. The proteins don't coil like the DNA does. The filaments slide.
So then, will they cross the M line when they?
They will cross the M line, and we'll see how, and that's hard to appreciate depending on the picture that we're looking at. If we were looking at this picture, for example, you say, well, how do they cross the M line? I see the M line as one continuous line. How do they cross it? But then if we go to another picture that's a little more detailed, we see that the M line is actually not continuous, but actually it is. So there's an M line, but we see that part of it wraps around myosin. And where actin crosses the M line, it's in front of and behind the plane of the board in areas like this. We'll get to all of that. I was just trying to give you a quick answer. Okay, so the filaments slide. So let's pick up Roman numeral 10, called changes in bands upon contraction, and we'll use this picture for that. Again, Roman numeral 10, changes in bands upon contraction. So let's start with the A band, which remember the A band, we've got labels up there. The A band goes from one end of myosin to the other, just as you can see here. A band goes from there to there. Does the A band change? It does not change, because the A band goes from one end of myosin to the other. Myosin doesn't shorten, so the A band is unchanged. All right, we've got an H zone within the A band. The H zone, this area, or that area, or that area. What happens to the H zone? It shortens, okay. We'll start there. But now, I want to go beyond that. The absolute number you pick is arbitrary, but what's the distance between the two marker caps? A
1 foot.
We'll start there. Now what's the distance?
6 inches.
Now what's the distance?
1 inch.
Now what's the distance?
Over here.
Okay, now what's the distance?
9.
0. Now what's the distance?
Negative.
Negative? Negative, did you say? That's what happens to the H zone. The H zone becomes negative. And now we also have the I band. What happens to the I band? and it approaches zero. It doesn't literally become zero because we have that protein that I told you not to worry about, and you still don't have to worry about it. What did I say the name of the green protein is? Titin. You can see if you compare this to this, it's like a little shock absorber to keep the Z line from literally butting up against myosin.
You didn't have that with your muscles when you contracted them, you were pumped about it.
Let's just say yes. You have it, so. And I don't know of a disorder where titin is missing. Okay, so now, how do they slide? As we look at different pictures from your book, and we'll start with this one. No, it's too big. So we don't see anything on myosin. It's just a line, thick versus thin. Myosin thick, actin thin. But as we go through these pictures in your book, they very subtly add something to myosin. What do you see here that you didn't see in the last picture?
Little fingers.
You see these little things, little fingers, little protrusions, little buds, little projections. They're actually heads. They're myosin heads. And what's going to happen, as bizarre as it is and as crazy as it's about to get, these little heads bind actin and ratchet. And every time these little heads ratchet, they are pulling the actin filaments in. Yeah, literally. These little heads, they bind, ratchet, release. Bind, ratchet, release. Bind, ratchet, release. And they're working together perfectly synchronized. But now let's expand that idea. Perfectly synchronized. If every one of these heads bound, ratcheted, and released at the same moment, what would happen when they were all released?
It bounced back.
It would bounce back. It would elongate. So they're perfectly synchronized, but every other head is in and out of phase with the adjacent head. It's like if we take a field trip and we're on a great big boat with no motor, out in the ocean, in the wind, and we're rowing. So there's 10 of us on the left and 10 of us on the right, and we're rowing. Well, when my oar is in the water and I'm pulling, you're in front of me and you're behind me. When my oar's in the water and I'm pulling, your oar's out of the water going back to the front. Then when your oars are in the water pulling, my oar's out of the water going back to the front. So we are 180 degrees out of phase, but perfectly synchronized. Because if all of the heads let go at the same time, the thing would, what would happen when you're trying to lift something? You wouldn't get anywhere. To appreciate the heads and the binding, we need a lot more magnification. So back to those pictures in your book where we follow the arrows. So we're taking a myosin myofilament and bringing it down here and blowing it up. So this is a myosin myofilament. These little bud things, whatever we call them, 5 or 6 different things, now we see them in detail. We see the M line and we see the true nature of the M line, and we can appreciate how the actin filaments can pass right through it. They're not really passing through it, They're in front of it and behind it. We take one molecule of myosin out of the myosin myofilament. We follow the arrow down here and we blow up that single myosin molecule. We see that the single myosin molecule has a tail, a neck, also called a hinge, and a head. This little arrow and this dashed line is showing us how that head can swivel back and forth. And that's this bind, ratchet, release, bind, ratchet, release. Now we need the detail of actin. Once again, I ask you guys, try to appreciate 1,000 years ago when people just cut bodies open and tried to figure out how they work. They didn't get anywhere. They had it so wrong. They used to think that liquid moved through arteries and gas moved through veins. It was hundreds and hundreds and hundreds of years before somebody ever correctly described the circulatory system and knew about oxygenated versus deoxygenated blood. Okay, so now we take an actin myofilament. We bring it down here and we blow it up. We've got all these orange and yellow spheres. The orange and yellow spheres are the actual actin components. We see that associated with actin, we don't see it at all here, but associated with actin there are two regulatory proteins. Troponin or troponin and tropomyosin. Tropomyosin is, I guess that's green, I'm not sure, but it's this filamentous protein that sort of spirals around actin. And troponin is this little globular thing. We see it here, we see it here, 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 and here. You don't have to worry about nebulin. You don't have to worry about G versus F with actin. But if you look at one actin subunit, the yellow portion is the binding site. They're calling it the active site, same thing. So in other words, where on actin do the myosin heads bind? They bind the binding site. That makes sense. But as I try to wrap my brain around all of this, I notice that the binding site is blocked. The filamentous protein, tropomyosin, is blocking the binding sites on actin. How can these myosin heads, how can these heads bind the binding site when tropomyosin is blocking the binding site? You ever read the last chapter of a book first because you're dying to know who done it?
It's only with mysteries. Because
Another
I
one of
like to
those
think-
pictures from the other book. So we've got myosin, the neck or the hinge, the head. This is not a cross bridge. This is a head. The publisher knows that they're not going to fix it because the author of the book is dead. The book will never be published again. They are aware of the typo. So, myosin, neck or hinge, head, actin, the long filamentous tropomyosin, troponin. The binding site is blocked. The head wants to bind the binding site, but it can't. What has to happen to expose that binding site? Check this out. Calcium binds troponin. It shifts. It pulls tropomyosin off the binding site. the head binds the binding site and swivels immediately. Look at the angle through the neck. Compare the angle through the neck here to here. So as soon as calcium binds troponin, troponin shifts, that pulls tropomyosin off the binding site. The head binds the binding site and it swivels. I'm sick of saying swivels. It's called a power stroke. Bind, ratchet, release. Bind, ratchet, release. It is called a power stroke. So as soon as calcium binds troponin, troponin shifts, pulls tropomyosin to the side, exposes the binding site, the head attaches to the binding site, the power stroke occurs, and we see that by looking at the change in the angle through the neck. Bind, ratchet, release. Bind, ratchet, release. And what happens? The sarcomere shortens. The Z lines move closer together, obliterating the I band, the A band becoming narrower and narrower and narrower, and the A band finally becomes negative because the actin myofilaments don't run into each other and stop, they overlap. So, how does all this happen? What is the molecular basis for all of this? What is the molecular basis for the calcium? Where does it come from? How is it released? What is the molecular basis for the calcium binding troponin, pulling tropomyosin out of the way, exposing the binding sites on actin so that the myosin heads can bind actin? I'll tell you Wednesday. It is absolutely critical. It is critical that when you guys walk through that door on Wednesday, that you are right where you are right now, as though you just took a 5-minute potty break. Okay, it cannot be a 48-hour break and you didn't study this. Okay, you've got to come into this room Wednesday morning, bang, ready to go. Because we're going to pick up exactly where we left off, and it is wild. It is crazy. It's the coolest thing in the world. Welcome to physiology. But now tell me, I asked you this last week, isn't this more fun than memorizing the muscle chart? This is how we work. Okay, this is how we work. Remember, everything, whether it's basic functionality, whether it's disease process, whether it's pharmacology, this is how detailed it is. This is how you come to understand it. This is the coolest stuff in the world, guys. The absolute coolest stuff in the world. Embrace it. Love it.